Welcome everyone to TRANSCEND's second seminar series presentation. We are especially fortunate to have Dr. Holly Brown-Borg leading today's presentation titled Translational Impact of Hormone Signaling on Aging and Longevity, Lessons from Dwarf Mice. Dr. Brown-Borg received her PhD in physiology from North Carolina State University. She was a research associate in physiology at Southern Illinois University, as well as a research associate in endocrinology at the USDA ARS MARC. Her main research focus is understanding the pathways and mechanisms utilized by hormones that lead to delayed aging and less age-related disease. Dr. Brown-Borg is presently a Chester Fritz Distinguished Professor in the Department of Biomedical Sciences at the University of North Dakota School of Medicine and Health Sciences. She is also the co-lead of the Health Research Core of the TRANSCEND study, a 5-year NIH grant whose goal is to increase the workforce of clinical and translational researchers in North Dakota. It is my pleasure to introduce Dr. Holly Brown-Borg.
Okay, can you hear me? Okay? All right, great. Thank you all for coming. Hope you enjoy lunch. You can thank TRANSCEND for that. And I really appreciate Kent and the TRANSCEND team for inviting me to come and talk about translational science in the context of aging research is what I want to talk to you about today. And if you have questions along the way, please stop me. I'll try to scan the audience. And so I wanted to first talk about translational science just a little bit. These are a couple of figures that have been taken from the TRANSCEND website. And it just talks about the different levels of translational research between T0 and T4. And we're kind of operating in the realm of T0, preclinical, but I'll talk about how the steps, at least in the context of aging, are being moved forward towards the T4 stage. And so just a little bit of information. So you can make basic biomedical discoveries, discoveries at the bench in cells and things like that in non-animal systems. Perhaps it's considered preclinical. Many times those discoveries are then moved forward into animal models. We kind of work in animal models and go backwards, so you can go back and forth between those areas. And then once certain discoveries are ready, they can be translated or moved forward into humans, into clinical trials. So this would be considered the T1 phase as you transition that work into humans, into these clinical trials. And then when you take that data further, you can more broadly apply it to different populations. And then we can take those or apply those findings and look in the North Dakota populations in particular, and then that can be taken to look at more broad, more global or worldwide populations, and that would be considered T4. So that's kind of the translational science process. We also talk about bench to bedside. So that's very similar, taking discoveries at the bench, moving those into medicine to improve health at the bedside. Trench to bench is a description that takes the opposite effect, or does the opposite thing. So you're taking epidemiological studies, what the observations there, and then going back to the laboratory and trying to look at mechanism and that type of thing, so you can really understand what you're seeing much better. And then there's bench to trench, which makes sense. It's laboratory to population health. And so our work in particular really focuses again on preclinical, which is the T0 or moving towards T0 and T1 phases. And we use animal models. And so our animal models we utilize are used for lots of different reasons versus using humans to counter the human aspect. So we use animal models because they have shorter lifespans. We're studying aging, so we can look at aging processes over 2 years versus 80 years in humans. So you'd never, as a grad student, you would never get done with your work if you're trying to follow the aging process, at least longitudinally. You could do it cross-sectionally, but those studies aren't as, they're important, but we don't get as much information from them as when we look longitudinally. We can also model age-related disease in animal models, so they're really important to use because there's lots of different ones that can be, you can make animals either genetically or pharmacologically, or use environmental things. our aspects and model age-related diseases. And so there's lots of reasons to use animal models, and that's some of the stuff that I'm going to talk about today. So even though we're here, our thoughts are always looking at to improve health in humans. So our thoughts and our targets are on the other side of this. And just to back up a little bit in terms of aging populations, so you kind of understand where the whole world is going. We know that in 2024, there's over 8 billion people in the world. It's a lot. And there's 10% of those are over 65. And so there's really a huge need in order to understand these age-related diseases, perhaps. And instead of focusing on each individual disease, if you can focus on aging, you can perhaps delay the incidence or delay the age-related disease itself. The really colorful graph is just looking at the most populated countries in the world. So India, China, and the United States represent the countries with the highest populations. And then if we look at North Dakota in particular, there's almost 800,000 people in the state in 2024, which is shocking. Because when I moved here a long time ago, everybody said there was like 650, 700. So we really have seen some population growth, and they're expecting it to go over 800,000 in the next 5 years. But our average lifespan, or life expectancy, is 76.9 years, and that's gone down. So it was 79 years, like less than 10 years ago. So, and it's not just COVID, it was already falling. And so we really need to understand that in our state. I think that that's extremely important. We have 17% of our population that is over 65. So that's much higher than worldwide. And it's supposed to be over 22% by 2027, which is just a few years away. So the importance of doing aging research, I think, is really key for the state of North Dakota. So this graph I've used a million times. I think it really describes what our approach is. So our approach really is to extend health span, not necessarily to extend lifespan, but to extend the amount of time that you spend healthy. So if the end of these arrows was 80 years or so, normally you spend the first 50 plus years healthy without any comorbidities or age-related disease, but then those begin to appear, and we spend the last 20, 30 years with age-related disease, at least one, and it's usually multiples as you get older and older. However, if we can target aging itself and not just a single age-related disease, if we can slow aging, then we will be able to extend that health span here and have a very short period of age-related disease, perhaps, and maybe get an extension of lifespan at the same time. But that's not really the goal of what we're doing. We're trying to extend this health span piece. So we know from the literature that aging is the major risk factor for almost all age-related or chronic diseases, I should say. And we know that if we can intervene with the aging process, that maybe we can reduce the incidence of multiple chronic diseases at the same time. So I also show this often, so everybody goes to the internet or wherever, to your doctor perhaps, to the supplement store, and thinks they can go from here to here by taking hormones, perhaps antioxidants, all sorts of diets or combinations thereof. And that's what we think about all the time. And so that's, again, it's expanding that increasing health span. But scientifically, we ask questions like, what factors can we mimic from long-living mice that we can apply to humans to improve human health? So that's always the background or underlying question. So a little bit about the research process itself for those that are not doing animal research or at the other end, perhaps, of clinical research. So we had made a discovery several years ago that animals that have low levels of growth hormone or no growth hormone in their system, they live 50% longer, minimally, than wild-type mice or mice that have normal levels of growth hormone. And at the same time, there was a couple of publications that showed animals or mice that had really high levels of growth hormone lived shorter. So if the normal lifespan of a mouse is 24 months, the animals with super high levels of growth hormone were living to 12 months. Not very healthy healthily either. The animals that had no growth hormone or very low levels were living to three to 4 years, so 36 to 48 months. So a huge extension in life just by altering it in these animals itself, a point mutation in a gene. And I'll talk about that in a minute. So that's kind of shown here. So the males and females of these dwarf animals, they live much longer than the wild type male and female. So that's both the average lifespan, the median lifespan, and the maximum lifespan. So you start to ask a lot of questions about where is this happening, what is happening, how is it happening, and that's kind of that research process that you follow. And then you use, I gotta go this way, you use cellular biology, you can look at biochemical pathways, you can use various interventions like diets and pharmaceuticals, all sorts of different things to try to figure out the mechanisms that are behind what the observations that you see. And then you can ask other questions. Can you apply that same thing to other model systems, perhaps, and interventions? So that's kind of the thought process that we use behind our work. So what do we really know about growth hormone? So you have to go back and you can look at physiology textbooks and you can find that growth hormone has these wonderful effects of stimulating muscle building, muscle growth. It stimulates cartilage and bone growth and other organs, but it also has metabolic effects. So it has lots of growth effects, and that's where it got its name, but it has these metabolic effects. So it raises blood glucose concentrations. So it's a counter-regulatory mechanism to hypoglycemia. So if your blood glucose goes down, growth hormone goes up to stimulate more glucose production and breakdown of glycogen. Then it also causes fat breakdown. So I put these bodybuilder pictures here. Don't know if they use growth hormone or not, doesn't matter. But what it does is growth hormone increases muscle mass and decreases fat mass. So there's wonderful aesthetic effects of using growth hormone, but there's lots of detrimental effects to using it also. And I'll talk a little bit about that in a second. However, when we talk about aging, growth hormone decreases over time. So once you hit about 25 to 30 years of age in a human, growth hormone concentrations start falling. And it's probably important biologically because you don't have that drive for proliferation towards cancer, but it also has other effects in terms of metabolism. So lower levels of growth hormone with aging, muscle mass and strength also decrease with aging, fat mass increases, unfortunately, with aging, bone mineral density and insulin sensitivity, or your ability to process glucose, decreases too with aging. And those are really characteristics of frailty, which is what we want to avoid. And then I put up these two figures because this individual, he's busy working, exercising on his exercise bike, and he's probably of a similar age as this individual who's a bodybuilder at 85. She looks like this, and still winning contests and things. So you can go both ways, but we're trying to study the differences in these individuals and how growth hormone impacts the aging process. So one of the other questions is, where is this happening? So we know that growth hormone is produced in the anterior pituitary, and it's under other regulation from higher levels in the brain. So the hypothalamus produces hormones that stimulate or inhibit the secretion of growth hormone from the anterior pituitary. And then growth hormone itself acts, its growth effects are primarily through the liver by stimulating IGF-1 or insulin-like growth factor production, but it also affects other tissues, as I mentioned to you on the previous slide. And so part of that discovery process in research is to look at mutations and figure out if there's mutations that are present both either naturally or genetically engineered, perhaps, that along the pathways of interest. And so we know that for growth hormone, there are mutations that are present that you can study in animals that affect the release of the hypothalamic hormone that stimulates growth hormone. There's receptors that you can study for growth hormone releasing hormone. You can reduce growth hormone or increase growth hormone itself. You can change the receptor so that it binds less efficiently or not at all. And you can also alter IGF-1 levels. So all these different levels of growth hormone, the growth hormone signaling pathway can be altered, and that can help you understand the process that you're interested in. So this is just a table that you don't need to read closely at all, but these are just characteristics of multiple animal systems that are out there that have a mutation in the growth hormone or IGF-1 pathway. And the thing to, what I just mentioned is you can impact at the hormone level, the upstream regulator, the receptor itself, or at the downstream effectors, such as IGF-1. And what we see is if you reduce, my arrows didn't come out. Anyway, there are arrows all over the place in here. But anyway, what you see is when you reduce signaling through the growth hormone pathway itself, you get an extension of lifespan. And these are all mouse models that are shown. They're impacted by different pathways within, or different factors within the growth hormone pathway. So this column here would show arrows going down. So plasma growth hormone is low until you hit this dotted line, which typically goes all the way across the screen, but not today. And so animals that are mentioned, or that are shown below there, actually have higher levels of growth hormone because their receptor is mutated. So the growth hormone is high, but it can't act. So you still have a reduced growth hormone signaling in that case. Stress resistance is increased, so those arrows are up in that particular column. Insulin sensitivity is decreased in all of these different animal models, whether they show growth hormone deficiencies or growth hormone resistance. So it just shows that there's multiple animal systems out there that we've looked at and others have looked at that impact the growth hormone IGF-1 signaling pathway, which results in an extension of lifespan. So just a little closer look at the animal that we've looked at most closely, the Ames dwarf mouse. They're deficient in 3 different hormones. Their lifespan extension is increased 50 to 70%, so that's males and females. They have that increase in stress resistance and an increase in insulin sensitivity. So the lifespan is great, but then our lab and other labs have looked at all of these other parameters in terms of aging systems, cells, organs, et cetera, and have found that they all are delayed or absent in these animals. And that delay leads to disease avoidance, if you want to kind of think about it that way. So we've looked at kidney aging, you can look at the immune system aging, cataract development occurs much later in these animals, fatal disease occurs much later, cancer incidence is almost 0. There's very little cancer in the dwarf until they get very, very old. We've already mentioned glucose homeostasis, learning and memory loss. These animals have very youthful levels of learning and memory in comparison to wild-type controls. So all those factors together are impacted by just lowering growth hormone. The opposite is seen in these growth hormone transgenic mice. So the ones that only live to 12 months of age, they're much larger than their wild-type counterpart. They don't quite look like this because we don't see them standing on 2 legs. But we see all the opposite things. So they have short lifespans, they're pretty unhealthy, and they have early senescence of multiple age-related, or multiple organ systems. So the question becomes, we're talking about translational science, so what about humans? So there's a lot of evidence in humans, but we have very variable systems. I mean, and we live all over the world in different environments and things like that. Our genetic background is hugely different. And so we do know that if you increase growth hormone pathologically, it accelerates aging in humans and it shortens lifespan. There's no question there. Growth hormone administration itself causes arthralgias, so arthritis-like symptoms, edemas, causes diabetes or high glucose because it causes glucose to be produced and released. It also causes cancer. So there's lots of impact of having or increasing growth hormone levels beyond what they're supposed to be, or later in life when they're actually falling. And there's also several papers that have showed if you reduce growth hormone signaling, kind of like what we see in our mice, the Ames dwarf, that they've shown that the secretion pattern is linked to longevity. Deficiencies are coupled to protection from cancer and are coupled with extreme longevity. If you delete the receptor, small population of individuals, but individuals that have a deleted receptor live much longer, the males do. If there's a defect in the receptor itself, it provides protection against cancer and diabetes, which are huge. And then there's one word missing here. Multiple health and longevity benefits have been coupled with IGF-1 also. So if you decrease signaling again through that growth hormone pathway in humans, you get extensions in longevity, but extensions in health span for sure. So this kind of, this figure just shows that evolutionarily this is also true. So if you're looking at lower species on the ladder, such as Drosophila or C. elegans and yeast, they don't have growth hormone. They have insulin and insulin-like growth factor, and that serves the same functions as growth hormone. And if you alter those pathways in these lower species, they live much longer. And then when you look at mammalian systems, so you're looking at the mouse, non-human primates, and humans, growth hormone is the master regulator of growth and metabolism. And if you lower that, you get an extension of lifespan and an extension of health span. And this is just an example of a human population. This is a group of individuals of short stature that live in Ecuador. And this researcher has studied this population of individuals for over 30 years and has found no type 2 diabetes. So they have a receptor, a defect in the receptor itself. So they have high levels of growth hormone, but they just can't respond to it. So again, they have reduced growth hormone signaling. So they have no type 2 diabetes. They're very insulin sensitive, which is great. They have higher levels of body fat because growth hormone causes lipolysis or breakdown of fat. So even though their BMIs average over 30, they still don't have diabetes, and they don't have any cancer. They don't necessarily live longer because it's partly environmental, and I can get into that later. But they don't necessarily live longer, but they live as long as their relatives that have normal levels of growth hormone. And this is just showing you the levels of IGF-1 and IGF-2, which are under the control of growth hormone, are much lower in these individuals. And this is showing the percent of diseases with the relatives that are in black and the growth hormone receptor deficient individuals that are in the white, which is not showing up. So there's no incidence of cancer, and there's no incidences of diabetes in this population, which is pretty amazing. So just to take a step back a little bit, and we talk about aging itself, and there's been multiple publications that talk about hallmarks of aging, hallmarks of health, that type of thing. So I'm going to go through a little bit of that just to put this all in context. So as I've mentioned, there's been decades of research, basic research done in the aging field. And what they've defined is a multitude of molecular processes that contribute to aging in different tissues and cell types. Those are all listed here. Deregulated nutrient sensing, which is where the growth hormone pathway would fit in. Loss of proteostasis. There's epigenetic alterations. All of these act together. They're all interconnected, and they influence one another. They're just not independent of each other. And they just represent many fundamental biological processes that all contribute to age-related disease. So in terms of health, when we talk about health span, and that's the direction that we want to move our own work, and the field wants to improve health span, we usually define health as a lack of pathology, right? So, or the absence of pathology. But here, they look at health more as a bunch of different features that are changing constantly that maintain physiology. Michelle, physiology, and Amanda. And if you disrupt any of these dynamic features or the physiology or perturb homeostasis, then you're going to, it results in broadly pathogenic aspects that occur that kind of derail the system. So if we can look at homeostatic resilience or oscillations in hormones and things. These are all things that are involved in maintenance of homeostasis, your responses to stress. So if you perturb any of that, you're going to perturb health. And so that's kind of the broader view when we look at past the hallmarks of aging. So what I've been trying to tell you all along is that these longer lifespans have to do with a slowing or delaying of aging-related processes. And if we can lower, or if we can slow down those aging processes, we can avoid lots of chronic disease. So the field kind of wants to look at aging in particular and not stop studying, but reduce the amount of emphasis put just on cardiovascular disease or neurodegenerative disease, kidney disease, that type of thing, because we're just looking at one single system and they're having impacts all over. So if you look at aging itself, then you can probably impact most of those diseases at the same time. So along with the field, they're busy looking at biomarkers. So we're always trying to find some sort of marker that we can use to evaluate what's going on over time. So this is just a whole bunch of different biomarkers that have been studied and are currently under study to try to determine if we can do more cross-sectional studies with humans, because we can't do longitudinal very easily, and figure out where they're at in terms of their aging process. And that kind of leads us to some of the interventions, which I'll talk about, the interventions that have been tested in both cellular models, in rodents, in non-human primates, and now also being tested in humans. So if we go back to the growth hormone example, we know that reduced growth hormone signaling reduces age-related disease. And these are all the mechanisms that growth hormone acts through, that decreased signaling, to increase longevity. So it decreases insulin secretion, it increases adiponectin, which is anti-inflammatory. It decreases peripheral IGF-1, it decreases cancer, and a lot of this is acting through the mTOR pathway. And so these are all, again, interacting, interconnected things that you can look at, or aspects of aging that you can look at that all lead to this increase in longevity. So we know that mice live 40 to 70% longer by reducing signaling, and they have less age-related disease. I showed you that it's very evolutionarily conserved from yeast through to humans, and we know that growth hormone decreases naturally with age. So the question is, when do you need to decrease it? How much do you need to decrease it? And if that's going to be impactful in humans. And we don't really know yet. So there's no really therapeutics to reduce growth hormone signaling with age at the moment. But some of the other interventions that are super popular right now that are being studied are one such intervention is calorie restriction or dietary restriction. So many of you have probably heard of this. Sometimes it's called intermittent fasting or alternate day feeding. There's dietary fasting mimicking diets, they're low-calorie diets that mimic actual fasting. Amino acid restricted diets, all of these different diets reduce the amount of calories going in overall in comparison to a normal diet. And in humans, there have been lots of animal studies here over 90 years that have shown that this definitely works in almost all mouse models, or mouse strains, but not all. But in humans, they've done a couple of different studies. One was called the calorie study, and they showed over 2 years individuals that they were supposed to be reduced by 25% their caloric intake. At the end of the study, it was actually only 11% because some people aren't that compliant, but it's really hard to do, but they reduced, because it was an everyday thing. So it wasn't every other day or that type of thing. So it turned out to be an average of 11% overall. But all of those individuals had decreased fat mass, increased insulin sensitivity, an increase in that hormone called adiponectin that really is anti-inflammatory. You want to have high levels of adiponectin. Their lipoprotein profiles were back to normal. They were really in the ideal place. And they had a decrease in many risk factors, serum risk factors for cardiovascular disease. So that was all very positive. But that was a 2-year study done in a few hundred individuals. And there's other studies going on. There's been multiple big studies in non-human primates. So NIH and the University of Wisconsin-Madison ran big studies with non-human primates that started in the mid-'80s, and NIH still has 2 calorie-restricted monkeys left. They can't cheat because they're given their food. So they have 2 left that are, I think they're 42 years old at this point. So for a macaque to be 42 is pretty amazing. But anyway, The University of Madison study is done, and theirs was slightly different, but they were still reducing calories. And other people are studying mimetics of dietary restriction. So who wants to really cut back on what you're eating when you could probably take a pill or something? It would be so much easier. I'm looking at Sarah. but so that's what people are kind of moving towards. They're trying to figure out if there's some sort of mimetics that we can mimic the biology of reducing that caloric intake. So there's that kind of have your cake and eat it too, that type of thing. And these are some of the molecules that are being studied. These have been shown in mice to extend lifespan. And so, they're still under study, and some of those have gone into clinical trials. And then we can also look at a bunch of metabolic regulators. So you want to reduce mTOR, reduce nutrient signaling, fool the body to think that it's, basically fooling it, what's happening. And you can do that by altering mTOR, AMPK, which is in the same pathway, and other molecules. Another intervention is metformin. So metformin's been around for a very long time. It was approved in France in the 1950s for diabetes. It wasn't approved in the US until the 1990s. But what metformin does is decrease the production of glucose from the liver. It increases the sensitivity of the muscle, in particular, to insulin. So those are both really good things. And it also decreases senescence-associated secretory factors, which I'll talk about in a couple of minutes. So these are really important aspects of metformin that have been shown to increase lifespan, at least in mice at this point. And so they've tried to set up a clinical trial, and I say tried because it hasn't happened yet. But the specifics of the clinical trial is called TAME, or Targeting Aging with Metformin. It's the first FDA-approved medicine or factor that can be used to treat aging. So it took many, many, many years to get the FDA to approve something to treat aging, because they only treat disease. And it's because it's medical-based, that type of thing. And so they finally got it approved through the FDA, which was a huge deal for the aging field. And then they set up this study, but it hasn't happened yet because they're waiting for the $50 million to do the study, which is what it'll cost. So they want to use enroll 3,000 older adults, put them on a 6-year placebo-controlled trial of metformin. And then they want to look at time to incidence of any age-related disease. Those are listed there. And then the primary outcome is to find whether there's a delay in gait speed, which is a huge indicator of frailty and aging in humans, or any other sort of disability in terms of mobility or cognitive function. They're going to look at markers biomarkers of aging. I'll talk about the xenomorphic piece in a bit, but again, it hasn't been supported yet because it costs $50 to $70 million. So they got part of the money from NIH set aside, but NIH won't pay for the whole thing. It's a lot of money, and especially now. So they're kind of waiting on this, but there's been lots of meta-analysis of people that have been on metformin for more than 20 years. Their cancer incidence is much lower. Their cardiovascular disease is much lower. There's all sorts of really positive effects. And there's a ton of people on metformin that are prescribed for metformin without being pre-diabetic or diabetic, because it's been shown to be fantastic in terms of delaying aging. Another intervention is called rapamycin. So this is a factor that was produced by bacteria that was found at Easter Island from the Rapa Nui Island. So got the name rapamycin. Rapamycin inhibits mTOR. So I was talking to you about mTOR earlier. It's a factor that regulates growth in animal systems and in humans, in the mammalian system. So this is the different things that mTOR does. It's involved in nutrient signaling, causes increase in fat mass. It causes growth of the beta cells that produce insulin, has all sorts of effects, but rapamycin blocks those effects. And what they've shown is that rapamycin increases lifespan in mice by 20 to 30%. And it slowed the progression, again, of cancer and neurodegenerative disease and atherosclerosis, which are huge, huge factors in terms of aging in humans. So there's been a huge clinical trial in dogs called the TRIAD, where they've got 40,000 dogs in the Dog Aging Project, and they're giving 1,000 of them rapamycin. It's ongoing right now. They're within this first year of rapamycin, and then they're going to do a 2-year follow-up. And the reason they're doing this work in dogs is because dogs mimic the environment of the humans that they're in incredibly. They start to look at all sorts of things that they really do. They're in that same environment. So they can do these studies in dogs and then apply that to the human work. The human studies themselves have already been in clinical trials, or rapamycin has been in clinical trials already, and it's been shown to improve immune function and physical function in humans. So there's a lot of real positivity with regard to rapamycin. There's some downsides of rapamycin, so they've now used an intermittent administration, and that's really reduced the side effects of this drug. And the final intervention I want to mention, they're called senolytics or xenomorphics. So as you age, your different organs and tissues accumulate senescent cells. And these senescent cells are still alive. They're not proliferating, but they do spew out these senescence-associated factors, which are cytokines that are pro-inflammatory for the most part. So they affect not only that cell, they affect all the cells around them, and they contribute to chronic inflammation with aging. So a lot of individuals have been studying these senescent cells and how you can reduce the number of senescent cells without impacting normal cells. So they've targeted them by genetic and pharmacologic approaches in mice. And they've been shown to delay, prevent, or alleviate lots of age-related disease by removing senescent cells. And so there's more than 30 clinical trials ongoing in humans right now. Most of them are phase 1 and phase 2. I think there's one phase 3 ongoing that are using these senolytics in humans to reduce age-related disease. And there's been some really promising results, mostly in lung fibrosis and arthritis. There's some studies going on with Alzheimer's and some other immune disorders. And then I've just listed some of these senolytics that are out there and available for use. And so the use of these senolytics will target aging itself, these senescent cells, reduce inflammation, and perhaps reduce chronic disease as a group versus individually in comparison. So lastly, to summarize, I just wanted to talk to you about translational research, where basic science fits into that in the context of aging. Our work is really at the T0. We're considered preclinical. We do some frailty work too. It's all considered preclinical, excuse me, because we're using mice. And the next step is to acquire, for us, would be to acquire human samples and human data to work with that. And we may be forced to do that here soon. Anyway, aging research really is quickly moving past that T0 stage finally, so it's very exciting for our field because of the numerous clinical trials that are underway. And the financial driver for anti-aging is huge. $52 billion market last year. By 2030, it's supposed to be $80 billion. So there's a lot of drive to reduce or to counter aging in humans. So we feel that if you can target mechanisms that cause aging and not individual diseases, that you can slow down or delay the appearance and incidence of many age-related diseases and perhaps transform medical practice. So slowing or delaying aging will prevent chronic disease is the underlying take-home message. So I just leave you with this. How old would you be if you didn't really know how old you are? And so it just shows an older nun playing with a children's toy. So anyway, thank you for your attention. If you have any questions, please let me know. And I thank again Transcend for the support to talk to you guys today and hopefully promote translational and clinical research in North Dakota. Archana. So, good question. So the first, people don't really measure growth hormone because it's all over the board all the time. So it's high at night, it's low during the day, it's high after a meal, it's high after exercise. So they measure IGF-1 because it's more stable and represents growth hormone's actions. And so people have measured IGF-1. And so there's been multiple populations, one in particular in New York, the Ashkenazi Jews, that have been studied very, very closely because they have a lot of centenarians, and they have lower IGF-1 levels throughout life. And so, and they studied them, their spouses, whether they're in the same population or not, and their children and children's children, and they're finding lower levels of IGF-1 in these families. And so there is some data, and there's some other studies in other populations that have also shown that that lower IGF-1 is related to longer life. Mm-hmm. Yeah, so there's been several different studies. So they've taken this Ames dwarf mouse and they've given back growth hormone during development and between 0 and 6 weeks of age. So they reach maturity, reproductive maturity at about 6 weeks of age. So they've tried to look at that window beforehand, if it's that congenital deficiency that's contributing. And if you give it between 2 and 4 weeks growth hormone, just for a couple of weeks, you get rid of a lot of the lifespan effects. So they still live longer, but not as long as they did before. So there's some sort of metabolic reprogramming that's happening, because if you give it after 4 weeks, nothing. There's been other people that have created mouse models that have normal growth hormone until at adult onset, so maturity, and then they drop the levels of growth hormone through some sort of a genetic manipulation anyway. And those animals don't seem to live longer, but they do live healthier. So their lifespan is similar, but they seem to live healthier. So there's something about reducing that drive for proliferation. And we think it's if you remove that drive for proliferation, then you're still taking in the same amount of nutrients and things, and it has to go somewhere, right? So all those amino acids that you take in and other nutrients have to go somewhere. So we think it shifts towards stress resistance and cytoprotection instead, because it's not being used to build big muscles and drive cancer, perhaps. So that's kind of the underlying thought behind reducing that you still take in the same amount of things, but you have to do something with them. So it's because we see this increase in stress resistance and cellular defense. So that's kind of the underlying thought behind that. So if you can reduce that proliferative impact or that proliferative drive, that you have to use those nutrients for something else. And it seems like it's cellular defense or promotes health, perhaps, by changing metabolism. Any other questions?
Holly, that was marvelous. That was really great. I really, really enjoyed it. You know, I'm sure you know this, but it made me think of the idea of prolonging health span rather than lifespan. You know, very old but famous poem by Oliver Wendell Holmes, the wonderful One Hoss Shay,
Mm-hmm.
which lasted for 100 years and a day.
Mm-hmm.
And then it fell apart, and it was like no disability, nothing first, just like a bubble when it bursts. So I'm wondering if there's a flip side to this. You know, for my money, if you could prolong my health span, really health span, and then just, you know, maybe checkout time is 2 o'clock. If I could check out quickly, that would be the best thing, because then you don't have that long period of disability. And I'm wondering if there are ways to sort of tweak this so that you could better term, pull the rug out from under longevity when it's done. So any thoughts about ending the health span quickly?
Mm-hmm. Two things. I heard that poem, a guy named Jay Olshansky is an aging researcher, he's an epidemiologist. And he quoted that a long time ago, and that's the first time I'd heard it, and it made a lot of sense, you know, this thing that lasts forever and then it just dies, it's just gone,
Right.
falls apart.
It's a famous Supreme Court justice, actually.
Secondly, I don't know. So there's been a lot of research. They've tried to figure out if this is like a programmed thing, and they can't really, you know, that doesn't seem to be programmed in any way, so that you could accelerate it at the end, you know, just to be done. That type of thing. People don't live. The oldest living person is 122, and there's been multiple people, and there's still multiple centenarians or supercentenarians that are called that are 115, 116, 118, but there's been no one that's made it past that point. So they don't know. That would be lovely if you could figure that out. And that's why I use that nursing home kind of thing, is that if you could live to 80 or 100, if you want just the last couple of years in the nursing home perhaps where you really needed some help, versus spending the last 20 with these multiple comorbidities that are happening along the way. You get one age-related disease and then another one pops up, then another one pops up, because they're all really quite related. So we don't know. I mean, that's what people are trying to figure out. That's why they looked at programming. They thought maybe it's programmed somehow into your cells. But you can get, in culture, you can get cells to double and double and double and double and double by avoiding certain things. You know, so I don't know. It's a good question. Yeah, exactly. Yeah, perhaps. If we all, you know, croaked at 100, you know, you just. Yeah, but that's why the approach, not the attack perhaps, is to look at aging itself and not those individual diseases, how the whole NIH was set up perhaps, you know, just because that was the early thoughts. Now, geroscience, it's called, we really want to study aging itself, because if we can slow that down, then in mouse models and in some human stuff, it already suggests that lifespan may not be extended, but you can certainly improve the health span by doing that versus individual drugs. And so but FDA, it was super hard to get that pushed through. And it finally got pushed through to treat aging, because aging is not a disease.
Right. Well, although somebody famous once remarked that old age is the only disease. So in that sense.
Yeah. So anyway, so that's kind of where the field has moved in the last 20 years or so. So any other questions? All right. Yep, go ahead.
Is there some certain cell types which are more sensitive or growth hormones or targeting those?
Certain types of what? I'm sorry.
Cell types, like those are like more sensitive for the growth hormones and targeting those
Certain cell types, yep. Perhaps there are certain cell types that are more sensitive just because if they have receptors and things for growth hormone, but lots of different cells have receptors for growth hormone that you may or may not think. I mean, immune cells respond to growth hormone. Hepatocytes, of course, respond to growth hormone. Myocytes respond. Cardiomyocytes respond. There's lots of different, and there's a bunch of different, I think astrocytes respond to growth hormone. So there's lots of different cell types. Neurons probably do too. So I don't think it's limited to just a couple of cell types personally. I think it's pretty global, but that's my bias. Yes? Mm-hmm. Yep.
I wonder what your thoughts on those approaches, and among many different approaches, what do you think would be most likely successful?
What do I take? No. That's always the question of aging researchers. What do you take? Resveratrol and sirtuins, you know, and the NAD modifiers. Resveratrol has been shown to work in obese animals in terms of lifespan extension and health span. If they're diabetic, it helps. It has positive effects. So that's more limited. That resveratrol is a substance found in red wine and grape skins and things. And you have to drink a lot of red wine to have those effects, a lot. not humanly possible. But the sirtuins is along the same line, so they alter SIRT, and it's been linked to aging by lots of different studies. I don't know if there's a really easy way to alter sirtuins at themselves, but NAD and there's a lot of nicotinamide ribosides and nicotinamide mononucleotides that are out there in the supplement world to take to raise your NAD levels, which is involved in metabolism. There haven't been clinical studies yet. There's a lot of anecdotal evidence, and there's a lot of products on the market. NAD, raising NAD levels, I see Barry going, no, no, no. But actually, there have been a lot of really positive studies in mice and stuff. There haven't been any studies in humans, but there's tons of supplements by very renowned people in the field that are using NAD, or using either NMN or NR to raise NAD levels, because they really think that it's underlying metabolism that needs to be changed in order to get these health effects. And so until the clinical studies come out, I don't know, I don't really know, but they are underway. So they are being used in clinical trials. But someone's making a lot of money off them. Any other questions? Great. Thank you for your attention. Appreciate it.